Good morning everyone. So I welcome you to the discussion crash course for your NEET exam. So since you have more time to prepare and you have studied the syllabus many times, so our aim should be now to go through the subject or the topics in a manner of learning rather than just theory. See, theory exam is like boards and all, where you write a lot, but by now that you have been preparing for your NEET exam, you know MCQ pattern is different. So the points have to be highlighted. I must have specific points pointing at the common questions asked, pattern of questions asked. So your notes and the discussion in class would be point format. As in where required, of course a descriptive account will be there, but then this is more of like a quick revision. So my advice to you would be, read NCERT and come, read your packages and the notes which we will provide to and come with your highlighted points, that these are the points which I forget, these are the points which are not clear to me. So when I am listing down the points and discussing the topic, then those doubts are cleared. If they are not cleared, of course you can ask simultaneously, right? So this is the pattern which we basically go with. So one, it will be point format. For you it will be easy to learn. The theory part, of course, the detailing, of course, can be done from the packages or the modules which will provide and from the NCERT, primarily NCERT. So you are supposed to read NCERT daily, read the chapter and come with the schedule which has been given to you, so that you are aware what we are discussing and you're also aware of your doubts and you're also aware where you get confused, so that those doubts, confusions can be solved. Is it clear? So we have that Telegram app where you can ask simultaneously. So let us see. So let us see what we are going to start with. We are going to start with the living world. A very basic chapter, but questions from which have always been asked. Some of the questions are overlapping or confusing. So let's just go through that part which is confusing. The first thing that we look at is how to distinguish between living and non-living. So we use terms like characteristic features and defining features. For us to define living, what we look at is the defining features. Defining features means they should be present in all living organisms and should not be present in non-living. So this is the criteria how I look at defining features. The first feature which NCERT discusses is growth, which can be identified by twin characteristics: increase in number of cells and increase in size. This is how growth can be looked at. There is an increase in number of cells and increase in size. Now if I look at this pattern of growth and I compare unicellular to multicellular, the main method of growth is cell division. In unicellular, if I look at cell division, then cell division will be responsible for both growth and reproduction. So growth and reproduction become synonyms or similar. In multicellular, however, there will be separate cells for growth and separate cells for reproduction. In multicellular, however, there will be separate cells for growth and separate cells for reproduction. If I now look at growth, so one reason why, if I now look at growth with respect to living and non-living, in living organisms, there will be always internal or intrinsic growth, always. That is growth from within. Whereas in non-living things, there is extrinsic growth, which we call apposition or accretion, that is by addition of material from outside. So intrinsic growth in living organisms may be a defining feature, but growth as such cannot be a defining feature. Growth will only be considered as a characteristic feature, means it is present in living. So what we can mark is that growth, intrinsic growth, is a defining feature for living organisms. Next thing which NCERT talks about is, next characteristic feature that we'll look at is the second characteristic feature that we look at is reproduction, that is ability to form young ones. Again, reproduction will occur by cell division. So again, in unicellulars the condition remains the same. Growth and reproduction are inclusive features. Growth and reproduction are inclusive features. Why? Because they can occur simultaneously. But for multicellular, they will be different. So exclusive. So one issue is this. Second issue with reproduction is that there are many organisms which do not reproduce. For example, worker bees are sterile. The queen and drones are fertile in honeybee. The queen, which is female, and drone, which is male, are fertile, but worker bees are sterile. Then there are humans which may be sterile due to genetic disorders or due to hormonal imbalance, which again means that reproduction may not be present in all organisms. So reproduction therefore again becomes a characteristic feature rather than a defining feature. It just remains as a characteristic feature therefore, but not a defining feature. The third characteristic that we take The third characteristic that we take is metabolism. Metabolism is the sum total of all metabolic actions which take place in the body. So metabolic reaction, a metabolic reaction, this is which is usually asked, can be done in vitro, that is in a test tube. You can do enzymatic reaction in a test tube, but metabolism can only occur inside a cell or organism. So whether it is unicellular or multicellular, metabolism will only be inside a living organism. So this is a defining feature. So this will become a defining feature. Now for metabolism to occur, we will require cellular organization. So cellular organization will also become a defining feature. The most complicated defining feature is consciousness. This is considered to be the most confusing and most complicated. So this is the most complicated defining feature. It is basically ability to sense the changes in environment, or ability to respond to stimulus. It is primarily this ability to sense the changes and respond to changes which all living organisms are able to do. Whether that response is slow or fast is different, but all living organisms, whether unicellulars or multicellulars, are able to sense the change and respond. So consciousness also becomes a defining feature. In humans, especially in humans, we take another defining feature, self-consciousness, to be aware of one's own body and changes which are occurring. So only for humans, you will also take consciousness. Of course, there are exceptions like patients in coma, but then there are exceptions under medical category. Under normal conditions, you can take consciousness for all organisms and self-consciousness especially for humans. Now, if you go through your last year's questions, you will find questions asking whether evolution is defining feature. No. The non-living things, your cell phones, everything is evolving at a fast rate. Evolution is a process. Whether memory is a defining feature? No. So memory, evolution, these things will not be put as a defining feature. So what are the defining features that we are looking at? 1. Intrinsic growth. 2. Metabolism. 3. Cellular organization. 4. Consciousness. And for humans especially, self-consciousness. This is what terms you are going to look for when you look at the point for defining feature. One thing which is common to all organisms is the genetic material. More or less same, there will be some differences. So all organisms have the same genetic material, that is DNA. So we'll say same genetic material, but of course with variations. This is something common in all organisms. Another thing we find in organisms is biological hierarchy. That is, cells aggregate to form tissues, tissues aggregate to form organs, organs aggregate to form organ systems. Organs aggregate to form organ systems, which will then form the organism. This is also called organization, which we study with respect to biological classification. Now you must understand there's a term we use, that the functions of tissues are not the functions of the cells. The tissue performs its function because of the aggregation. Individual cells cannot perform the function of the tissue. Individual tissues cannot perform the function of the organ, and therefore. So you have to be very clear that cells cannot perform function of tissues, tissues cannot perform function of organs. It's the aggregation which is responsible for the functioning, which means we have large number of organisms which we look at as biodiversity. If we look at biodiversity is the heterogeneity or the different organisms present and studied. So the diversity at the level of organisms, the different kinds of organisms that we have. So the organisms can be plants, animals, fungi, lower organisms, bacteria, everything included. Now if we go by IUCN, we have 1.7. So these are the values we use in biodiversity as well. In even biodiversity we use the same values. These are the organisms known to us. There may be many more. These are the ones which we have discovered till now, and there may be many more. So our aim is to study this biodiversity. Our first step of biology is to study and understand this biodiversity. So how do we study this biodiversity? Definitely we cannot study this biodiversity studying one organism at a time. There are 1.8 million organisms. So I cannot study each organism separately. I have to have a method of organizing them in groups so that common characteristics can be studied in groups and specific characteristics can now be studied for a unique organism which you want to study. So I need to therefore classify them. I need to understand their characteristics. What are the characteristics? Classify them, name them accordingly. Now to study biodiversity, we use two terms which are most confusing. Study of biodiversity. There are two terms which are used which are basically confusing for you: taxonomy and systematics. See, they are overlapping terms. So taxonomy are the rules and regulations or the tools and techniques which you use to study organisms. I'll take a very simple example. The rules of binomial nomenclature which were framed by Carolus Linnaeus is part of taxonomy. What characteristics to consider, how to name, what should be the criteria of naming, how to use the name further, how to modify the name if required. These are the rules that we use. That is taxonomy. Using these rules when we classify organisms, name organisms, so that an evolutionary history can be understood. The grouping has to be such that I can understand which is primitive and which is advanced when that evolutionary relationship is depicted during that characterization, classification, naming, identification. Then I'll call it systematics. So systematic arrangement of organisms. What is systematic arrangement? Evolutionary relationship to be depicted. So taxonomy is more of theoretical part. These are the rules and regulations or the tools and techniques used for characterization, classification, nomenclature, and identification. These are the 4 things which are integral part of taxonomy. These are 4 things which are integral part of technology, or the basic principles are related to these principles we devise: how to characterize, how to classify, how to name, and how to identify. So the rules that we set will be taxonomy. What is in systematics? We say systematics is the systemic or proper arrangement of organisms. What do I mean by proper arrangement of organisms? My only criteria of proper arrangement of organisms is to study the diversity and, this is now important, evolutionary relationship between organisms. So we say taxonomy plus evolution is systematics. This term is taken from systema, derived from term systema, that is systematic arrangement. Taken from systema, that is systematic arrangement. Now taxonomy can be now further, I can look at taxonomy as classical or modern taxonomy. Classical taxonomy, where I use primarily morphology, where primarily morphology is used as a criteria, but then this will not be able to give very good rules. So what we use today is modern taxonomy, in which we may use many characters such as morphology, anatomy, genetics, embryology. So several comparative features may be used. And more recently, Julian Huxley has introduced neo-systematics or neo-taxonomy. This has been introduced by Julian Huxley, where we use specific criteria to check classification. Where we use specific criteria, for example, we will do this in plant kingdom otherwise. Cytotaxonomy, where cell structure is used, or you may use numerical taxonomy, where comparison of many characters is used. Then we may have chemotaxonomy, when the biomolecules of organism are used for comparison. So we'll come to this later. Presently we are just looking at what is generally taxonomy and systematics. So using this taxonomy and systematics, our aim is to first characterize the organism, accordingly name and classify the organism, and then develop taxonomical aids so that it is easy to identify organisms. So our sequence will be first to name them, then to group them in the form of the taxonomical categories or hierarchy, and eventually devise aids so that we can study all this easily. So let me start with nomenclature. Let me start with nomenclature. Nomenclature is naming of organisms. Nomenclature is naming of organisms. We do have general or vernacular names. Vernacular names means names in local language. These are easy to pronounce, easy to use, easy to remember. But then the issue with these names is, the problem with these names is that these names sometimes have incorrect meaning. They may have incorrect meaning. They may be misleading. And sometimes one organism may have many different names, whether in same language or different languages. So it becomes difficult. To use these names for scientific discussions. Why? Because we will not know actually what we are discussing. So the need for scientific naming. So the need for scientific naming, which should not only be correct, but also it should be universally accepted and used. Now there are several scientists who have given these type of naming techniques and methods. What we follow is the binomial nomenclature. What we follow or what we use is binomial nomenclature given by Carolus Linnaeus. What does binomial means? Binomial means two. Binomial means two. This means that the name of species will have two components, a generic epithet and a specific epithet. This is something I would like to discuss, that there is a difference between specific epithet and species. Let us say the name of species is Brassica campestris. So Brassica is the generic epithet which can also be considered as genus. Campestris is specific epithet which is not defining any species. Campestris here is the specific epithet to define the species Brassica campestris. Have you understood this point? Do not confuse in an MCQ with respect to specific epithet and species. Brassica campestris on the whole is the name of the species, in which Brassica is the generic epithet which is same as genus. Campestris is not species. I will not say it is species campestris. I can say it is species Brassica. I can say it is genus Brassica, but I cannot say it is species campestris. I will say campestris defines the species Brassica campestris belonging to genus Brassica, because in this genus Brassica different species will have different specific epithets. Now Jansia now is defining species Brassica jansia, belonging to genus Brassica. We can have different Brassica oleracea. So this is like name and surname. This is the surname of your family. All of you will have the same surname, but you will all have different first names. So just saying the surname, then I am addressing the whole family. If I am just saying the surname, I am addressing the whole family. If I am just using this name, then I might not know which family's child am I talking about. So to define you, I will have to use your first name along with your surname. The first name can be reused. For example, first name means the specific epithet. I can reuse the specific epithet. For example, Agaricus. Agaricus is a different genus. It's a mushroom. Agaricus campestris. So I have used campestris again, but with a different genus, so it becomes a different species. So generic epithet cannot be reused for more than one genus. You can give a generic epithet only to one genus. Specific epithets can be reused. You have Mangifera indica, you have Oryza indica, you have Tamarindus indica. So indica is common, but the generic epithets will be only for a genus. The other rules you are already aware of, taken from Latin, so we underline them or put them in italics. The first correct name is accepted. A binomial epithet will have the scientific name. For example, Mangifera indica. It will be then followed by the name of author and the year of publication. So I may now write L-I-N-N as Linnaeus. Linnaeus, he gave this name in 1753. So name of author and the year. You will not underline Linnaeus in this because they are written in normal English. This is what is written in Latin. This in totality will be called the binomial epithet. Now binomial nomenclature is the most used and accepted. Lamarck then gave a trinomial nomenclature as well. So Lamarck suggested a trinomial nomenclature as well, where three names are used. For example, if I take for humans, what is suggested is Homo sapiens sapiens. So he suggested the use of subspecies as well. He suggested the use of subspecies as well. Tautonyms, these are terms which are usually asked, are when the generic epithet and specific epithet are same. When the generic epithet and specific epithet are same. In such a case, now this is what has been used in zoological nomenclature. It is no longer used, but in botanical nomenclature you will not have any such name. So where are these rules? Where are all these rules written, how to name, what to name? So we have to have standard books or standard texts giving us these rules. So what do we use for this? We use the standard texts or books for naming, that is International Code for Botanical Nomenclature, International Code for Zoological Nomenclature. So these are standard books, codes which are used, how to name, what terms to use. So these are the standards which we take for nomenclature. Now apart from this, trinomial is rarely used, tautonyms only in zoology, botany you don't use tautonyms. Then there are terms like synonyms if an organism has two names. Usually one name is followed as per the law of priority. Now the second contribution which Linnaeus made was to give us a hierarchy, a taxonomical hierarchy. Another thing which we gave, say this we have named, now we must group. Now we must group them. So next thing which he has contributed is to give taxonomic hierarchy. Next thing that we look at is the taxonomic hierarchy. Hierarchy means to arrange organisms. Hierarchy means to arrange organisms or arrange in order of increasing or decreasing similarities and dissimilarities. Now what is the aim of hierarchy? If I look at this species, it's one member. To a genus, several species will be present like Brassica rapa, Brassica jancea, Brassica oleracea or Panthera leo, Panthera tigris. So a genus may comprise of many species. Many genus will then form a family. So what we are doing is, according to their similarities and dissimilarities, we are grouping them. In taxonomical hierarchy, we are going to group organisms according to their similarities and dissimilarities. There are 7 obligate categories which we use. There are 7 obligate categories which we use. Kingdom, phylum or division, class, order, family, genus and species. There are 7 obligate categories which are used. Kingdom will be the highest category. Species is what is considered as basic or natural unit. Reason is that in nature actually species exists. The animals don't know their genus and their family and their order, no. In nature what exists is species. So what are these categories? We have just aggregated species. What are these categories? What exists in nature is species. These categories are aggregation of species with respect to increasing dissimilarities and decreasing similarities. Means the members of a species will have maximum similarities. They will have maximum similarities amongst themselves because they belong to the same species. As you move up, the similarities will decrease because the group is becoming larger. So this will have minimum similarities. So as I move from species to kingdom, term which we use is common characters. Common means commonly shared characters. Similarities. So commonly shared characters or similarities will decrease. Kingdom will have maximum diversity. So kingdom is going to have maximum diversity, maximum diverse. So plant kingdom will have algae, brio, pterido, gymno, angio. Animal kingdom will have non-chordates, chordates. So maximum diversity, and species will be minimum. So diversity is going to decrease and similarities will decrease from species to kingdom. So this is basically aggregation or grouping of species according to their similarities and dissimilarities. These are called obligate categories. Every category will have a group of organisms. Like if I say plant kingdom, then plants are the group of organisms. The group of organisms which belong to an obligate category will be called a taxon. Taxon is a biological group which belongs to an obligate or physical category. Taxon is a biological group which belongs to a physical or obligate category. So if I now say family Fabaceae, so all the members of Fabaceae will now be taxon. So the classroom which you have is your category. You are sitting in the classroom, you are the taxon. Year after year the classroom remains same, the classroom doesn't change, but the children in the class will change. So taxons can be different. So in a category family, if I look at category family, this is my physical category or physical taxon. The biological entity can be different. You can have family Fabaceae, you can have family Solanaceae, you can have family Malvaceae. So in the same rank, this is the physical rank, the biological members will vary according to the species that you are studying or according to the group of organisms that you are studying. Please be very careful here. A physical rank is a category to which the biological rank or taxon belongs. So you will place reptilia or you will place mammalia, any of these in the given physical category to which they belong. A category therefore can be occupied by different biological entities depending upon the species that you are studying. This is how we look at the taxonomic categories. So we are grouping the organisms into different categories. There can be of course sub-obligate and non-obligate categories like subclass, supraclass, subfamily, they can be there, but the 7 obligate categories are must. So let us look at these categories from species onwards. Why? Because the natural unit of classification is species. Why we start with species? Because it is the real or the basic unit of classification. Other units of classification are just aggregation of species. So species is the basic unit of classification. In MCQ I can write all the terms. Real unit of classification, natural unit of classification. Now several definitions have been suggested to define a species: a morphospecies, linear species, allochronic species, sympatric. So there are n number of definitions which have been given how to define a species. The most acceptable concept which we now use to define the species is the biological concept of species given by Ernest Mayr. Biological concept of species is based on the fundamental feature of organisms, that is reproduction. What do I mean by fundamental? Means it's the primary criteria how a species recognizes members of each other. A dog recognizes another dog because of its ability to reproduce. A dog knows it cannot reproduce with cat, so they are different species. So in nature, different species exist together because members of one species can reproduce only amongst themselves and not with others, which you call reproductive isolation. So according to the biological concept of species, which is the most acceptable concept, those members will be considered as one species which are capable of interbreeding. Members of a species, how will I define a species, or how will I look at a species? Those members which are capable of interbreeding amongst themselves to produce fertile offsprings. It is must, and this should be done under natural conditions, not under captivity. Humans have made all types of hybrids, but under natural conditions those hybrids will not be formed. So members of one species are not able to reproduce with the members of other species due to reproductive isolation. Now in an MCQ, please be careful. This is how I look at a species. Reproductive isolation is a mechanism used to separate different species. Reproductive isolation is not used to form a species. To form a group of organisms to be called as species, it is this definition which is used: ability to reproduce. The ability not to reproduce is a method how one species is differentiated from another. In an MCQ, you have to be careful. If I ask you a species is defined on the basis of, created on the basis of, I give you both the options and this is the correct answer. Species is created because of its ability to reproduce with members of same species. Reproductive isolation is a mechanism or device used to separate the members of one species from the other species, not to create a species but to separate it from other species. So in an MCQ, please be careful as to what language is being used. See, what is an MCQ? In a multiple choice question, I may just change not present, present. I may just change few words here and there and the meaning changes. So do not mug up these statements. Do not learn these statements without understanding, because you can just change the language of the question, language of the options, and the meaning will change. If you have learned a strict statement and you are looking for that strict statement in an MCQ, then you will not be able to reach an answer easily. You might reach an answer, but you may use more time. Time is very important in NEET. You have to work within the limits of time. So your terminologies should be clear to you. So this is how I am looking at species. There are several definitions which have been given, which you also do in zoology otherwise. So morphospecies, lineal species, etc. Now species will be defined as per the binomial nomenclature. The next group that you have is genus. The next taxonomical hierarchy that you look at is genus. Genus will be group of closely related members of a species. So genus may comprise of many species, like Panthera is a genus which may comprise of Panthera leo, Panthera tigris. Panthera now will be called a polytypic genus. It may comprise of only one species, like ginkgo is a genus which has only one species, Ginkgo biloba. So ginkgo therefore will be a monotypic genus. Ginkgo therefore will be a monotypic genus. So depending upon this, a genus may have many species. So Panthera leo, Panthera tigris, Panthera pardas, they all belong to same genus, so polytypic genus. Some genus may have only one species, Homo sapiens, Ginkgo biloba, then they will be called monotypic. Several genus will comprise together to form the next hierarchy level, which is family. In plants, in flowering plants, especially angiosperms. In angiosperms, family is defined on the basis of morphology. One of the criteria we take is morphology, both vegetative morphology and reproductive morphology. This is what your chapter of morphology is. Remember, we studied root, stem, leaf, flower, fruit, seed, and then we use all those characteristics to identify the angiospermic families. So families are then grouped together. So related families which have some similarities will be grouped together to form order, and orders will be grouped together to form class, phylum and kingdom. This is how we put the hierarchy. Now you should be very clear about the table given in NCERT, which tells you about different members, their species, their genus, their family. Like Mangifera is given. So for Mangifera, the class, order, family. Musca domestica is given. So class, order, family. Homo sapiens, there is a table given, which you will do thoroughly. Usually we ask that table in comparison, mark the odd one out. So I may give 3 families and 1 order. So odd one out will be the order. So you have to look at that in totality as to how do I group class, family, order, class, or if we start with phylum, class, order, family, genus and species for a given species, right? So this is our primary taxonomical hierarchy. Now our aim is to devise methods as to how to study this taxonomy, how to make it simpler, easier, more direct to study this taxonomy. For this we use taxonomical aids. For this we use taxonomical aids. The taxonomical aids are used, these are methods used or these are techniques used to study identification. These are mechanisms or methods used for study identification. They can be used for conservation as well. For example, for conservation, we use botanical gardens, we use zoological parks. Otherwise, we may use for preservation. Preservation is of dead organisms. So, I may use a museum, I may use a herbarium. So, taxonomical aid is not just to understand taxonomy and classification, but can also be used for conservation, preservation. It can also be used for display of plants and animals, like in museum we use it for display. Because by displaying or observing something, you are able to understand it better. So, what are the common taxonomical aids that we use? First thing that we look at is the herbarium. Herbarium is a collection of dried, pressed and preserved plant specimen, but the specimen will not be preserved in total. Parts are preserved. You cannot preserve the complete tree or the complete plant. Small plants can be preserved totally, but not large trees. So what we are going to do is, in a herbarium you would have prepared an herbarium in school also. First collection is done, you will clean them, then you will put them in a plant press, or at home you just put it in newspapers and then you press them. You press them properly so that all parts are fully visible. You allow it to dry. You can change your papers in between. In laboratory we can use mercury chloride for poisoning, so that there is no fungal or bacterial growth. So once it is completely pressed, dried, then you will mount it on a herbarium sheet. Once it is completely pressed and dried, you will mount the plant on herbarium sheet. So you are going to do collection, then you are going to do pressing, drying, and then mount it on herbarium sheet. Usually the dimensions and label of herbarium sheet is asked. There are different sheets we use. The standard sheet we use is 41 centimeters by 29 centimeters. Of course, you can convert it to inches as well, 16.5 or 11.5 inches. So you are going to put your plant, you are going to display your plant here in the center so that all parts are fully visible. Minimum overlapping parts should be there so that they can be easily studied. In the corner we are going to put a label. Here we are going to put a label size of 12 centimeters by 7 centimeters. The label is going to have 7 things. The label, we put 7 things. Scientific name, English name, common or vernacular name, name of family, place of collection, date of collection, and collector's name. A question which has been asked again and again, whether time of collection is taken or not. Time means in hours and minutes, that is not taken. Date is of course taken. Date of collection is of course taken. But time with respect to number of like at 28 hours, 24 hours or 16 hours, no, that is not taken. So this is what we'll put in the label. Additional description can be put on a separate sheet, but on a herbarium sheet you will just put this much. If there are larger parts, then they can be put in jars with formalin. Now we group our herbarium, or we classify our herbarium. The herbariums in India have been classified according to Bentham and Hooker classification. In India, the herbarium is grouped and classified according to Bentham and Hooker's classification. The primary aim of herbarium is going to preserve parts of plants, no doubt. But if I am asked the primary aim of herbarium is, then it is quick reference. You can go to a herbarium, refer to a herbarium, and look at the type of plants, the shape of plants, so that you can easily study. The largest herbarium in India is the Central National Herbarium in Kolkata. Otherwise, across the world, it is the Royal Botanical Garden in Kew. The second taxonomical aid we can use is museum. In a museum, we have preserved specimens of both plants and animals, preserved usually using formalin. We can use acetic acid also, but usually using formalin. This is usually called FAA solution. So they are preserved in glass bottles so that they are easily visible. So they are used for display. The main aim is display for easy observation. Display for easy observation. Large animals can be stuffed. Large animals can be stuffed. Then we have botanical gardens. In botanical gardens, we grow plants, especially exotic plants, breeders varieties, which we have created by plant breeding. We can grow flowering plants which have distinct flowering pattern, fruit bearing. Then some plants which are endangered. So we can use botanical gardens. We can use botanical gardens for display, for observation, for breeding and conservation. Same as zoological park, or what you commonly call zoo. Same is for the zoological park. in which we usually keep the wild animals for conservation, breeding programs, and of course for visiting. You enjoy visiting a zoo to look at the animals which you normally cannot see in your day-to-day life. This will be the zoological park. Then we have keys. Keys are used for identification. Keys are analytical in nature because they are based on comparative features. They are analytical in nature as they are based on comparative features. In a key, what we will do is, we will take two contrasting traits. This pair of statements which gives you two contrasting traits is called couplet. Couplet is a pair of statements. So couplet will be two statements taken together, a pair of statements with contrasting traits, with dissimilar characters or traits. For example, I may take a couplet. Let us say I am looking for a bird or something. If I am looking at that, I may say beak present, beak absent. So this pair is a couplet. Then I will say if beak is present, go to statement 1, go to statement 2, couplet 2. If beak is absent, go to couplet 10. It is like a treasure hunt. This is my first couplet. So if beak is present, go to couplet 2. If beak is present, then this becomes the lead. If beak is present, but if beak is absent, then this will become the lead. Means out of the two statements of couplet, each statement can act as a lead depending upon the characteristic of organism. So first we'll make couplets on contrasting or dissimilar characters. Each couplet has two statements, opposite different statements. So the statement of the couplet which correlates to the characteristic of the organism that you are studying, that statement will now be called the lead. Lead will take you to the next couplet. Next couplet similarly will have two contrasting statements. Let us say if I go to couplet 2, I may then be asked feathers present, feathers absent. If present, go to couplet 3. If absent, go to couplet 15. So if feathers are present, then this will become the lead. One lead at a time will take you to the name of species. For each taxonomic category, there will be a separate key. You cannot identify the kingdom, phylum, class, order in one key. No. First, you will use a key to identify the kingdom. Within the kingdom, then you will use a key to identify the phylum. Within the phylum, you will use a key to identify the class, and so on and so forth. So there is a separate key for every taxonomic category. A single key will not take you directly to the species. So there is a separate key for every taxonomic category. That is why we say keys are analytical in nature. Then we can also use reference books. Reference books such as Flora. Flora is a detailed account. Flora is basically a detailed or descriptive account of the habitat, and in that habitat distribution of plants. It does not describe the plant as such. It may give small descriptions for identification, but flora is primarily an account giving you an index of plant in a given habitat and how those plants are distributed, pattern of distribution of that plant in the given habitat. Now if I want to identify a plant, then I'll use a smaller account, manual. Manual is more like a handbook. It will help you to identify the plant on the basis of the description. It is like a handbook which will give you the description of plant, not the habitat, for easy identification. In identification, naming is included. If I want to study one particular taxon, I will use a monograph. Monograph will be used. A monograph will be used for description and listing of one taxon only. Taxon means it can be listing of family, order, class, species, family, anything, but for one taxon only. A catalog is basically arranging the plants in alphabetical order. An alphabetical order of arrangement of plants, that will be then called a catalog. So this is our basic chapter of living world. When you go through NCERT, there are of course terms and everything, but what we have done is the most common terminologies and points on which questions are based. So you will read through this, mark your doubts in questions for tomorrow's class. So we are going to start with now the next chapter, biological classification. It is basically a continuation of what we are leaving here. Why? In this chapter we have talked about naming, we have talked about taxonomical categories, but then we have not discussed how to create those categories, especially the kingdom. So let's look at biological classification. Let's look at biological classification. So let me start with classification. Classification is grouping. Classification is grouping of organisms on the basis of similarities, dissimilarities, and what is considered very important today is the evolutionary relationship. Now, if we are using only few characters, if I am using only few characters to classify, I will call it artificial classification. where I am using one or few characters only. Natural classification uses many comparative characters. Natural classification uses many comparative characters. But what we follow today is the phylogenetic classification, in which the primary criteria is, or I may say, the classification should be such that it is able to depict the evolutionary history. Now, if I look at classification, my first aim is to group them according to the highest category kingdoms, and then I may create rules or criteria to form phylum, class, etc. So when I classify, or when I look at this process of classification, my first aim is to form kingdoms. So let's look at the highest category. What are the kingdoms in which we have classified organisms? So kingdoms of life. First such grouping where kingdoms were created was the two kingdom classification. This was given by Carolus Linnaeus. He has grouped organisms into just plants and animals. The common criteria he used was cell wall, which is present, absent, mode of nutrition. He considered all plants to be photoautotrophic and animals as heterotrophic. Locomotion absent, present. Then contractile vacuole. This is how he has studied. I am not saying this is absolutely correct. This is what he has studied. Absent, present, and response to stimulus. He says absent, present. This is his criteria. How he has grouped plants and animals into two kingdoms, plant and animalia. Now this is an artificial system. Then came along the three kingdom classification. The three kingdom classification was given by Haeckel, where he has introduced kingdom Protista. His criteria of identifying kingdom Protista was unicellulars, not unicellular eukaryotes. When Haeckel created kingdom Protista, he just separated as many unicellulars as possible from both plant and animalia. And second criteria he took was they should be mostly aquatic. So he left several unicellulars here because they were not aquatic. So his primary criteria was unicellulars and aquatic. So he gave the kingdom Protista. Rest of the animals and plants were still under kingdom Plantae and kingdom Animalia. So kingdom Plantae and kingdom Animalia still had unicellulars. All unicellulars were not removed because all are not aquatic in nature. Then came the four kingdom classification. This was given by Copeland He then created kingdom Monera as a kingdom of prokaryotes, and most of the prokaryotes or all the prokaryotes which you know of are unicellular, but his main criteria was prokaryotes. So kingdom Monera became a kingdom of prokaryotes. Kingdom Protista, which was already existing and was accepted by Copeland, became a kingdom of unicellular eukaryotes. The remaining multicellulars were then kept in Plantae. So Plantae had fungi as well and Animalia. Now what we accept today or what we use today is the 5 kingdom classification given by R.H. Whitaker. What we use today is the 5 kingdom classification given by R.H. Whitaker. He has used 5 criteria for classification. He has used 5 criteria for classification. So the criteria for classification, accordingly, he has kingdom Monera, kingdom Protista, fungi, Plantae and Animalia. So the first criteria he has used is complexity of cell, where this is prokaryote and all others are eukaryotes. And all others are eukaryotes. Then he has used complexity of thallus or thallus organization, where Monera and Protista are unicellular, fungi is multicellular, with loose tissue-like organization, which you call basically hypha or mycelium. Plantae are also considered as multicellular, usually with tissue level of organization, and Animalia are multicellular. The level of organization may vary from cellular aggregation, as you find in sponges, to tissue level, as you find in hydra, to organ system level. So most evolved or thallus organization you will find in Animalia. Third, reproduction. Please note, it is not mode of reproduction. Reproduction here primarily talks about method of sexual reproduction, which is absent here. In Protista it is present, wherein you can have gametic meiosis or zygotic meiosis. Both are present. In fungi again it is present, where we usually have the zygotic meiosis. In Plantae it is either zygotic meiosis, as you find in algae, or sporic meiosis, which you find in all other plants, Brio, Pterido, Gymno, Angio. In this case we have only gametic meiosis. In this case we have only gametic meiosis. The next criteria which he has taken, the next criteria that we look at is mode of nutrition. You will have the highest maximum modes of nutrition here, varying from auto to heterotrophic. Autotrophic to heterotrophic. In Protista also we can find both auto and heterotrophic, but Monera you will find more modes. Fungi is heterotrophic, primarily saprophytic. Plantae will be photoautotrophic only, means photosynthetic. Animalia is heterotrophic with the main mode being Holozoic. The fifth criteria which is taken is evolutionary history. Or relationship. Then we explain the evolutionary history or relationship. Then we say that Monera is the most primitive, which came from the progenote. They gave rise to 3 types of eukaryotes, that is kingdom Protista. They gave rise to 3 types, that is Protistan algae, slime molds, and protozoa. This is kingdom Protista. Protistan algae gave rise to Plantae. Slime molds gave rise to fungi, and protozoa is precursor of Animalia. So these 3 together will be kingdom Protista. This is how we arrange. This descriptive or diagrammatic arrangement to show evolutionary history is called a cladogram. This diagrammatic arrangement to show evolutionary history is called cladogram. Okay, so what we'll do is, we'll stop here today. We'll continue with Whittaker's classification, that is the drawbacks, etc. And we'll look at the 6 kingdom classification and then Monera, Protista, other kingdoms in the next class. So please revise and come, read up the chapter and come, the previous one and the next tomorrow's topic as well, so that you are thorough and the revision is done well. So take care. We will meet tomorrow then. Good day.